that brings together these 3 ovarian cancer organizations, and more importantly, created a foundation to bring together researchers that really had never worked together, and this is what they've done across all the various DREAM teams. And that I was well aware of Dr. DeAndrea's work, admired it a great deal, but had never spoken to him personally when he called me and said, you know, I'm interested in, you know, forming a DREAM team and applying for Stand Up to Cancer. And, you know, we met in person the first time when we went to Philadelphia to defend our proposal to the Stand Up to Cancer Review Committee. And I think it's just an example of how it brings together people who wouldn't otherwise be working together. And you have this opportunity to sit down and say, if I could work with anybody in the world, who would I pick to work with on this idea? And that's kind of what we did. We put our heads together and we you know, picked out the people that we thought would be the most productive and fun to work with in this environment. And so it's really exciting for us now to have this opportunity to get started. So we have this, our project is called DNA Repair Therapies for Ovarian Cancer, but you're going to see that it's more than just about therapy, that we have a big prevention aspect of it. So what is DNA repair? So DNA repair is a basic process that's really essential to all cells and all organisms because it's necessary for cell survival, because it fixes errors that happen during replication. And it also fixes damage to DNA that happens from external agents, such as sun or chemotherapy exposure or other things that happen to a cell, even when it's not replicating. And there's a type of DNA repair called homologous recombination. And sometimes we just say HR. And HR is the most important type of DNA repair for repairing breaks in DNA. And when DNA damage is not repaired well, it leads to cell death. So we know that ovarian cancer really has few druggable targets. And what that means is, you know, when you've read about new drugs being used for melanoma and targeted drugs being used, ovarian cancer doesn't have a lot of specific mutations that lead to obvious targets for biologic drugs. But one thing that's kind of common to ovarian cancer is this defective DNA repair. And ovarian cancers that have certain types of DNA repair are sensitive to a class of drugs called PARP inhibitors. And major advances now in the basic science of DNA repair also allow us to propose some novel ovarian cancer drug therapies and hopefully also improved risk assessment for ovarian cancer. So those are going to be the 2 themes that I talk about. So this is sort of a structure of our DNA Repair Dream Team. We want to impact ovarian cancer mortality through novel therapies and through cancer prevention. And as far as I know, this is new to Stand Up to Cancer, that there hasn't been a big cancer prevention trial in one of the other DREAM teams, to my knowledge. And so we have two thrusts. One is therapy and one is prevention. And then advocacy is really important to this effort, both the support that we're receiving from the different foundations, but advocates that are on our team and are going to inform our trials for therapy and prevention, and then also help us get the word out as we get data out from our trials, get the word out to the community so that this can be a faster process than sort of what is typical. Stand Up to Cancer wants things to happen quickly, as hopefully do we all. So this is our organization of our DREAM team. I'm a co-leader with Dr. DeAndrea, and then we have an executive committee. There's 6 different institutions involved, and each institution has a lead person. We have and then we have basically these 3 different categories of science: cancer genomics and DNA repair, developmental therapeutics, and then behavioral science and cancer prevention, and then a lot of subcategories under that. And these are all being brought together across our different institutions. And so the institutions that are involved are Memorial Sloan Kettering, Dana-Farber, University of Washington, University of Chicago, the Mayo Clinic, and MD Anderson. And then we also have a lot of collaborators in industry, as well as a collaborator at the NCI. So we have three aims of our proposal, which is the first aim is really involved with kind of understanding the science of DNA repair in ovarian cancer and using that understanding to predict who will respond to different kinds of therapy. And so that science is sort of the foundation for the team, and that was sort of really what got us excited about bringing together this team in the first place, is we'd get some of the leading DNA repair experts from around the country together who hadn't previously worked together, and we would use that science to inform our second aim, which is about therapy for patients who already have ovarian cancer, and then our third aim, which is about risk detection and cancer prevention. So I'll go into each of those in a little bit more detail. And then from each aim 2 and aim 3, the therapy and the prevention, we will be collecting samples which we will then use to inform our science again, so that it's really a circular effort. So aim 1 is the hardcore science aim, so I'm not going to torture everybody by going into this in incredible detail. But the rationale for the aim in general is that 50%, we know about half of high-grade ovarian cancer have deficiencies in that HR DNA repair pathway. And recently, one of the PARP inhibitors, olaparib, was approved for use in ovarian cancer for women that have BRCA1 and BRCA2 mutations, which are important genes in the DNA repair pathway. But we also need to identify additional women who would benefit from PARP inhibitors, and we need to learn to sensitize ovarian cancers that don't have BRCA mutations to PARP inhibitors and also to chemotherapy. So we're going to look at other aspects of DNA repair and figure out how to manipulate that or use them as predictors for different types of therapy. So the idea is that a better understanding of DNA repair alterations will lead us to have improved biomarkers that allow us to pick the best therapy for patients. And then we'll also learn how ovarian cancers develop resistance to various therapies. And they do that by altering their DNA repair response. And that can help us, again, lead to more effective therapies. And down at the bottom, I just have sort of listed a lot of the people that are involved in this basic science aspect of the proposal. So the summary of aim 1 is that we want to develop tests to identify various DNA repair deficiencies. We're going to relate DNA repair deficiency to therapeutic outcomes. We're going to identify alterations in DNA repair that lead to resistance to therapies. And then we're going to test our new understanding in the context of our novel clinical trials. So that's aim 2. And then we're going to develop ways to break down DNA repair in cancer cells and make them more sensitive to therapy. So even ovarian cancers that don't have issues with DNA repair and are sort of inherently sensitive, we're going to figure out ways to make them sensitive. So now maybe moving on to something hopefully that's of interest to this crowd is development of novel therapies. So that's the aim 2. And in aim 2, we have 3 clinical trials that we're going to carry out. And the rationale for all these clinical trials is that ovarian cancers that have intact homologous recombination or proficient HR are not sensitive to PARP inhibitors. And even cancers that have BRCA1 and BRCA2 mutations that are initially sensitive to PARP inhibitors ultimately develop resistance. And they do that usually through restoration of DNA repair. So we want to develop new drugs that inhibit DNA repair and then sensitize the cancer cells to PARP inhibitors so that they can work together. So the idea is to take ovarian cancer cells that have intact DNA repair, make them defective, and add a PARP inhibitor, and then that's like a double whammy. So we have 3 different ways that we're going to do that in these 3 different clinical trials because we don't know what's the best way to do that. So we're going to kind of test several different strategies. So in the first clinical trial, we're going to use a drug with a PARP inhibitor called dinaciclib as the second drug. So this is a drug that blocks homologous recombination repair, and it has been shown in cell cultures and preclinical models that it confers sensitivity to PARP inhibitor to cancer cells that are not otherwise sensitive. And you can really enhance in mouse models the response of, if you combine these 2 drugs, dinaciclib, and in here the PARP inhibitor we're using is veliparib, which is an AbbVie PARP inhibitor. And this already is in an ongoing phase 1 trial. So a phase 1 trial is where we figure out what the right doses of the drugs are. And in that phase 1 trial, there's been a patient who had a very remarkable response who had a triple-negative breast cancer. And then the phase 2 study that we're going to do is going to be all women with ovarian cancer treated at the ideal dose. And then this is kind of the schematic for what the study will look like. Women with advanced ovarian, fallopian tube, or primary peritoneal cancer who haven't had a PARP inhibitor before will be randomized to either get just the PARP inhibitor by itself, or they'll get the combination of drugs. And if they get the combination, and then the idea is to see if the combination of drugs is better than just the PARP inhibitor by itself. But to sort of enhance that understanding, women that only get the PARP inhibitor, if they don't have a good response, they'll be allowed to move to the other arm of the combination, so that everybody could have the combination eventually if the single agent wasn't working. And then we're going to be we will be doing biopsies and looking at the science of it during this process so that we can predict who really benefits from this type of approach. And that study will be run probably predominantly at the Dana-Farber and then maybe at one other institution. And then the second trial is a PI3 kinase inhibitor combined with a PARP inhibitor. And this is based on the idea that 30% of ovarian cancers have hyperactive signaling of the PI3 kinase pathway, which is not a DNA repair pathway, but it's been shown that inhibition of that pathway can cause a DNA repair deficiency. So a phase 1 study has already been ongoing at the Dana-Farber and came from a previous Stand Up to Cancer grant that was focused on the PI3 kinase pathway. So an oral PI3 kinase inhibitor leads to decreased DNA repair. And so far, 44 patients were enrolled in this phase 1 study. And the dose-limiting toxicities, meaning the things that really impacted patients, were liver toxicity and depression, which was significant. So responses were pretty good. 27% of patients that were really heavily pretreated had a good response, but the toxicity was pretty high. So so recently in that phase 1 study, we've changed to a different PI3 kinase inhibitor that's less toxic, and we're still combining with the same PARP inhibitor. This time it's olaparib. And on that new combination, 6 ovarian cancer patients have been enrolled to date, and there's been one very good response. So that is sort of the idea, is to take this. We're still dose finding for this combination, but once we have the dose the maximum dose identified, then we'll do a similar study to what we just described with that combination, where patients are randomized to either the single agent PARP inhibitor or the combination. And again, we'll allow crossover if the patients getting the single agent don't get a good response. And we'll look at biomarkers both for DNA repair, but also for the PI3 kinase inhibitor pathway, and figure out who are the patients who are going to benefit from this type of approach. So then the third study is earlier, meaning we haven't already started dose finding for this combination. It's going to be a brand new combination, and it's looking at another DNA repair inhibitor drug called in a different gene called ATR, which an inhibitor of ATR can enhance killing of ovarian cancer cell lines in combination with a PARP inhibitor as well. So we're going to look at this combination first in just mouse models to look at toxicity and how effective the combination is, and then in the second part of the grant we'll be doing a phase 1 trial, which is just the dose finding. So this is really earlier in development, but I think it's kind of an exciting opportunity. Yeah, I'm not going to go into that in more detail. So then the third aim of the grant is really to look at risk assessment and prevention. And so this is based on the idea that 20% of ovarian cancer is caused by germline mutations in ovarian cancer genes that include BRCA1 and BRCA2, but about 4% of the time is a different ovarian cancer gene than the typical BRCA genes. But, and we know that risk-reducing self-pinch oophorectomy, in other words, removing surgically the ovaries and the fallopian tubes, is effective at decreasing mortality in high-risk women. But genetic testing is really underutilized, and unfortunately, genetic testing is often done after someone gets cancer, in which case it's a little bit too late for prevention. It really helps family members more than it helps that individual avoid the cancer. It's also evident that not all women are willing to undergo a risk-reducing self-pinch oophorectomy prior to menopause because they want to avoid the negative impact that surgical menopause has on them physiologically. So this aim is sort of geared towards assessing all these components: risk assessment and surgical prevention. So the first part is we're just going to really sequence a lot of samples for our known ovarian cancer genes that we've already identified. We've already tested blood from about 2,000 women with ovarian cancer, and we have 11 ovarian cancer genes that are listed here that include BRCA1 and BRCA2, but 9 other genes as well. And we're going to test an increased number of women without cancer from a study called the Women's Health Initiative, and we're going to increase our number of cases to 4,000. And this will allow us to generate more precise risk estimates for women who carry mutations in these genes. And we may identify a few new ovarian cancer genes along the process. So that's kind of really the first part of the aim, is to really solidify our definition of ovarian cancer risk. And then the second part of the aim is basically a clinical trial that's called CheckMate, and that stands for Chance to End Hereditary Ovarian Cancer by Making Testing Better and Easier. And the goal of this study is to develop a new paradigm for genetic testing that would increase access to women for risk assessment. So barriers to widespread risk identification right now include the fact that the identification of target patients by physicians is poor. So identifying patients who are at risk of carrying an ovarian cancer gene right now is not done very well by physicians. There's a lack of availability of genetic counselors. There's a lack of knowledge by both physicians and patients about the opportunity for genetic risk assessment. It's in our current paradigm, which requires going and visiting a genetic counselor to get blood drawn and counseled and then coming back to receive results, is inconvenient. It's not available to many women outside of major cities, and it's expensive. So we want to sort of try to, make that more accessible by creating a totally new paradigm and then assessing that paradigm for how it works. So what we're going to do is enroll several thousand women who don't have ovarian cancer but are at risk for having an ovarian cancer gene. So these are women that have either a personal history of breast cancer or some kind of family history of either breast cancer or ovarian cancer. Also, we're going to enroll women who have already have a relative that have a known mutation. So we call that cascade testing. Cascade testing is where somebody in the family has a mutation, like a BRCA1 mutation, and all first-degree relatives of that individual can get tested, and if they test positive or negative, they're at 50-50 chance. If they test positive, then their first-degree relatives can get tested, and so on, and you can expand within a family out the genetic risk assessment and give people more accurate information, and that's currently really underutilized in our current system as well. So people will come in and basically through a web-based portal. So what patients will enter the study won't ever have to travel to our study sites. They'll be able to be enrolled from anywhere in the United States. They'll get to talk to someone on the phone, but they'll sign an electronic consent. They'll fill out baseline questionnaires. They will be sent a saliva test kit in the mail. And then our partnering company, Color Genomics, will sequence our ovarian cancer genes, and results will be disclosed to the patient. They'll be able to receive genetic counseling, but it will occur over the phone. And our primary outcome is to identify how many people complete the entire process and receive their genetic results. Like, are we making this easier for patients by bringing the testing to them in their home and not making them come to us? And then we're also going to measure the satisfaction with the web-based genetic services, their knowledge and risk perception, psychologic distress, and how they use that information. Do they communicate it to family members? Do they, if they are identified with an ovarian cancer risk gene, do they utilize that to take prevention strategies? And this is the company that we're partnering with, and the really reason that we're partnering with them is because they've already developed a web-based platform that that we're going to be able to basically co-opt for our study, and then add a lot of questionnaires and things that will allow us to assess how it's working for patients. So then finally, our last study, our third part of our prevention is we're actually going to have a surgical prevention study, and we call it WISDOM, for women choosing interval salpingectomy with delayed oophorectomy to postpone menopause. And this is based on the idea that getting your ovaries out decreases the mortality for women with BRCA mutations, but it has a negative impact on health, sexual function, and well-being. And 80 to 90% of ovarian cancer in women that have BRCA mutations we know arise in the fallopian tubes. And possibly just taking out the fallopian tubes could be useful to decrease the risk in sort of two high-risk groups of women. Those who've completed their childbearing, but they're really younger than we would tell them that they need to get their ovaries out. So they just want to get their fallopian tubes out for now and their ovaries out later. Or those that really, where we are recommending that they have their ovaries and fallopian tubes out because they're already at an age of risk, but they're delaying it because they're anxious to avoid menopause. And we feel that in that situation, a salpingectomy may be better than doing nothing at all, which is what some women are choosing. And so FORCE, which is a advocacy group called Facing Our Risk of Cancer Empowered, did a survey of high-risk women, and 34% of women were interested in salpingectomy. So a pretty high fraction of women are interested in just having their fallopian tubes out, even though the current recommendation is to have both the ovaries and the fallopian tubes removed. So MD Anderson, one of our Stand Up to Cancer sites, is on schedule to complete enrollment of a pilot trial of 40 women who are given the option to choose between salpingectomy or getting their ovaries and fallopian tubes out. And they are almost done enrolling those patients, so it's been actually well received. So the enrollment for this study will include women that are aged 30 to 50 who are premenopausal and they have a known mutation in an ovarian cancer gene. And we'll provide them sort of scripted counseling on the recommended age that we believe they should get their ovaries out, but they will self-select. So this is not randomized. They self-select to either a risk-reducing salpingo-oophorectomy, which is the standard procedure that's done, or an interval salpingectomy, meaning only having their fallopian tubes out, and then a delayed oophorectomy at some point down the road. And we will have a data safety monitoring board that will monitor whether there are cancers that occur in either group of women, and we'll clearly stop this trial if we think that cancers are arising in the women that are delaying having their ovaries out, because that's you know, we don't want to endanger women, but some women are choosing this option already, so it's really important that we gather this information to determine if it's a safe option. The primary outcome really is going to be looking at sexual function, and the reason for that is that we know that going through a surgical menopause can impact sexual function, and it's important, and it's one of the reasons that women want to delay menopause, and so it's something that we can measure and see if it's different between the groups. We'll also look at menopausal symptoms, psychological symptoms, and then pathological results, like if any cancers are identified at the time of surgery or later. So basically, we think that we can decrease ovarian cancer mortality, not just through therapy, but through prevention. And we can develop really a new paradigm for ovarian cancer prevention that really addresses all three areas: defining the risk, a genetic risk assessment that makes it more accessible to patients, and then surgical prevention that potentially increases option for women that are not happy with our current options. Then we can get maybe more women on board with prevention if we give them more options. So advocacy in this process is really critical. Advocacy is important both for our clinical trial design, for recruitment, to help us interpret results, and then to publicize results to patients and providers. So we really are looking forward with working on this aim with our advocates to make it successful. And this, I'm not going to pause here for too long, but this is just sort of the whole DNA Repair Dream Team is supposed to be a 3-year project, and this is our timeline for how we're going to get all of our aims accomplished in that amount of time. And I can guarantee you that Stand Up to Cancer is going to hold us totally accountable, so it better happen. So just in summary, the strengths of our repair team, these are the 6 institutions, again, that are involved. And we have a diverse complementary team of investigators from 6 world-class institutions. We have novel therapeutic interventions to deliver near-term patient benefit. We have the potential to reduce mortality through prevention. We're going to apply new basic science mechanisms to DNA repair profiling. And we have biopsies from clinically annotated PARP inhibitor trials that we'll be able to use. The recent approval of a PARP inhibitor sort of has spurred everybody's interest in this area. We have many industry collaborators that are going to work with us on this proposal and therefore get therapies to patients hopefully more quickly. And we have a committed group of advocates that I know are going to be important in making this successful. So again, this is just like where I started. Basically, our DNA repair Dream Team sits on a foundation of science, of investigators from many institutions that are going to be working together. Therapy and prevention are both going to be equally important in our Dream Team. So that's really it.
Thank you, Liz. Liz, you may want to leave your presentation up.
Oh, yeah, I meant
just in case anybody has any questions.
And nobody wants to look at my email.
Thanks, Dr. Swisher. We do have time for some questions. Just remember, you can submit them in your Q&A box that's there on the left-hand side of the screen. So we have started to get some questions in. So this is sort of a general question, and you did kind of address this, but I thought it's maybe worth repeating. So the question is, how do you feel the Dream Team is really different from any other large research grant?
So it has to do with this collaboration of people from different institutions, some of the leaders in the field, and also from leaders in different fields. So Alan DeAndrea is a pediatric oncologist. He had never worked in ovarian cancer before, but he is a DNA repair expert. A pediatric oncologist and a gynecologic oncologist aren't brought together every day together. So, even in a big NIH grant, you're generally trying to bring together maybe 2 institutions at most. This is sort of like we sat down and we said, if we could just pick anybody we wanted, who would we try to get together on this particular problem? So that's what kind of makes it unique. And another thing that makes it unique, honestly, is like when you create a huge consortium and you do it through the National Cancer Institute or some big fundraiser like that, there's a big bureaucracy associated with that. There's a lot, and Stand Up to Cancer really cuts through all the bureaucracy. They have a timeline that is very tight, and everybody just has to jump on board and figure out how to make it happen, and there's a whole lot less dithering that can happen. So I think the timeline is one thing that makes it unique because they want things to happen quickly, and the second thing is just this being able to bring together people who wouldn't have otherwise even thought about working together. You just sort of say, you have this opportunity to be like, hey, there's this Stand Up to Cancer team. Who could we put together that would be, difference makers? So that I think that makes it really fun for us, that opportunity.
Okay, that's great. Thank you. How can people are asking how they can enroll in one of the clinical trials you just described?
Well, they're not open quite yet. So the Stand Up to Cancer team is, we don't even have any money yet, so it's starting July 1st, so it's right around the corner. So what we're doing is gathering everything together to be ready to start as soon as we can. But even then, like our genetic study and our prevention study, it's going to have to, right now we're working on finalizing the protocol, and then it has to be approved by the Human Subjects Committee. So it's probably going to be, I would say, we're hoping that we'll actually be able to enroll patients in about 6 months. There's a lot of work that needs to go into it between now and then for the prevention pieces. But hopefully we're going to try to move as quickly as we can and be enrolling, towards the end of the year. And then the therapeutic studies right now, because the first 2 studies, they're phase 1 studies, they're both being run at the Dana-Farber. And if somebody wants to get on one of those studies, they can look on clinicaltrials.gov, and they can search they can look at what's available for ovarian cancer patients at Dana-Farber. That's where they're run. If anybody's trying to to find the trials and they can't, I'm sure I can give that information to Sarah and we can get that out. But those are still in the phase 1 part, but they're pretty far along. So the number of patients that you enroll in a phase 1 study are limited because you are looking for dose finding. So you before you can raise to the next dose level, you have to be sure that there wasn't too much toxicity. So you enroll like 3-4 patients at a time, and then you wait and make sure they're tolerating it before you can enroll the next group of patients. So that sometimes is frustrating for patients because all the slots aren't open all at the same time.
Right. And somebody asked this question. So presumably patients from other institutions can go to a participating institution to see if they could qualify for one of these trials.
Right, definitely.
Okay. Right.
Yeah.
Okay. Let's see. So I think with the first trial, you mentioned fallopian tube cancer. And someone asked if would someone with fallopian tube cancer be eligible for all of the trials, potentially?
Oh, yeah, they are. They are eligible for all the trials. The eligibility is basically the same across all the therapeutic trials. So fallopian tube cancer, primary peritoneal cancer, and ovarian cancer, and more of those 3 cancers are being lumped together in clinical trials. It probably is somewhat arbitrary. Some of the things that we call ovarian cancer probably came from the fallopian tube. Same goes for peritoneal cancer. So we think that those 3 cancers are very closely related. So any of those 3 cancers will qualify for any of the trials.
Great. Let's see. Can DNA, this I guess is a little general question, can DNA repair therapies be used and are they useful for recurrence with metastasis?
Yeah, so those clinical trials are really going to be all patients that have recurrence and do have metastases. So yeah, we think they are useful in that setting. Ultimately, it may be that we use them more as first-line treatment, but like many new therapies, we're testing them to see, if we can help women that have recurrent ovarian cancer and have metastatic disease.
What applicability will all this research have for those of us in remission is a question.
Well, so the prevention piece, I think, may help keep your daughters safe and your sisters safe from having to go through what you've already gone through. So that's one piece. The second piece is that hopefully you never need another therapy because you're in remission and you stay that way. But if your cancer ever did come back, then what we want is that we have the best treatments available and we can do tumor testing and figure out, not like to hunt around for therapy for someone to see what, that you have X% a chance of, you have a 30% chance this treatment's going to work. Well, we want it to be, as close to 100% as possible. We want to be able to predict which are the best therapies for an individual cancer. So hopefully for someone in remission, that's not an issue in the future. But we definitely want to keep as many therapeutic options for patients as possible.
Okay. Here's a good question. How will you address women's breast cancer risk who are found to have BRCA mutations when you do the genetic testing?
We actually, for the Stand Up to Cancer presentation, we really simplified it and made it just ovarian cancer, but that's a really great question, is that many of these genes that we're testing for impact both breast and ovarian cancer risk, and patients will get counseling about both breast and ovarian cancer risk. And any other cancers, if they have, say, a mutation in a gene that causes ovarian cancer but also causes colon cancer, we give the gene-specific information for them. And we're even going to be testing for more than just the 11 ovarian cancer genes. We're going to actually be testing for 19 genes that include other breast cancer genes too, because many of the patients coming into the trial, that genetic testing trial, will have had breast cancer already. So we want to tell them whether or not they're at risk for ovarian cancer. That's kind of one of our main goals. But obviously they're kind of interested in what might have caused their breast cancer, even if it doesn't impact their ovarian cancer risk. So we're going to test them for all the really well-accepted breast cancer genes as well.
Okay. Great. There's some questions coming in, and I'm wondering if you can just sort of comment in a general way about how PARP inhibitors might help women who are BRCA negative.
So yeah, so ovarian cancers that have BRCA mutations are the ones that have been most studied with PARP inhibitors, but it's clear that for women that don't have BRCA mutations, that there are ovarian cancers that respond very well to PARP inhibitors. And those are cancers that have, like BRCA1 or BRCA2, they have defects in genes like BRCA1 or BRCA2 that also impact DNA repair. So they create the same sensitivity that a BRCA mutation does. So there's a subset of ovarian cancer that behaves like it has a BRCA mutation even when it doesn't have a BRCA mutation. And we are working right now to figure out exactly what's the best way to identify those patients so that we could say, who amongst people that doesn't have a BRCA mutation should we treat with a PARP inhibitor that will behave like a BRCA cancer would have behaved. So we're running a clinical trial, not through this Stand Up to Cancer, but I've already basically been involved in running a clinical trial that is called Ariel 2, where women are having biopsies, and we're figuring out who amongst women without BRCA mutations are responding to PARP inhibitors. And then we're going to use that information going forward to inform our trials and other trials.
Okay, great. Let's see. This may be a little overly specific, but there's a question, do you know if there are any olaparib trials for non-BRCA positive patients? To my knowledge, not olaparib trials. So because olaparib was the first drug that came to market, the company sort of had the option to just go for the best responders, which in the easiest ones to pick are those with BRCA mutations. But every other PARP inhibitor that is being developed for ovarian cancer has trials that are open for women without BRCA mutations. So that includes rucaparib, which is the Clovis drug, niraparib, which is the Tazaro drug, and veliparib, which is the AbbVie drug. And each of those companies has clinical trials running that include women without BRCA mutations, but they're in different settings. So some are for women who are having their first treatment, newly diagnosed cancer. Some are for women with recurrent ovarian cancer. Some are for women who have not had very many treatments before. Some are for women who've had at least 3 treatments before. So the specifics on which trial is eligible for varies, but if you look at clinicaltrials.gov and you put in ovarian cancer and PARP inhibitor, you get a list of a bunch of the trials. It shows like where they're open, whether, who's eligible for them. So it can give you enough basic information to tell you which trials that you might want to call about and get more information about, especially if they're in your area. Also, I just want to jump in. Dr. Swisher is mentioning clinicaltrials.gov. On the OCRF website, and the other organizations may have it on their website as well, we have a clinical trials finder.
Good.
And it uses all the information that's on clinicaltrials.gov, so you'll find the same results, except the interface is a little bit nicer. It allows for a more personalized search. It just makes it, clinicaltrials.gov is very comprehensive, but a little unwieldy.
It's hard to
sometimes.
wade through, yeah.
Yeah, exactly. So a question came in, if women eventually become resistant to PARP inhibitors like they do to, say, platinum drugs, how do you think that research will address this?
Well, so that's a great question. That's really something that we're very interested in solving. So one is with these combination of drugs that I mentioned that we're doing in AIM-2, where you have a drug combined with a PARP inhibitor. That first drug kind of knocks down the DNA repair in the ovarian cancer and allows it to be more sensitive to the PARP inhibitor. So that's one strategy that we're actively pursuing. Another way is like using basic science to better understand how different cancers become resistant will give us insight into ways to kind of overcome that. So it's not the same for every ovarian cancer, that the ones that develop resistance do it in different ways, but there are some common themes, and the more that we understand it, the more that we can kind of figure out how to keep that from happening or to overcome it if it does happen.
Okay. Okay. Thank you, Dr. Swisher. I think we will wrap it up there. So I want to thank Dr. Swisher for an excellent and informative presentation, and I want to thank everybody for calling in today. I also want to thank the Ovarian Cancer National Alliance and the National Ovarian Cancer Coalition for working with us on this event. And thank you to Genentech for making all of our webinars possible. Two things I want to say because questions have been coming in about this. Yes, this webinar presentation is being recorded, and the archived version will be available after the presentation. So you will all get a link to that presentation or to the archived version probably tomorrow. And someone else asked me to reiterate, not clinicaltrials.gov, but on the OCRF website, on the home page, if you go to Clinical Trials tab, you'll get a link to our Clinical Trials Navigator. Maybe I'll see if I can include a link to that in the follow-up email that you'll all be receiving as well.
Yeah, send that to me. Send that to me too.
You do it? Okay.
Yeah, because I need to be sending my patients to that instead of clinicaltrials.gov, which is difficult to use. So thank you for bringing that up.
Yeah, no problem. And the nice thing about the Navigator is it actually will only show you like trials that are still enrolling, and it's a little better. So do take a minute to fill out the survey that will appear on your screen at the end of the webinar. It's very helpful to us. And that's it. So thank you all, and have a great day.
Thank you.